the old slides.
You're good.
Thank you.
Thank you.
So this we'll be covering some work that actually both Adam and I and others worked on a few years back. And what's great about this work is it was able to be implemented on a recent clinical trial that Medtronic just released, and Adam will go into detail on that. But it's important to realize this was a long effort over, around 10 years worth of work. So with that, maybe Adam, next slide. So I just want to acknowledge the work is really a collaboration between tons of sponsors. We've got like Eric here, who was gracious enough to ask us. We have a lot of collaboration with the FDA. And we also worked closely with MDIC, and Adam will discuss what the MDIC is. And we have internal colleagues at Medtronic that were also heavily involved. So just a big family of people. So what we're going to cover today, we'll outline it as we're going to talk about some of the challenges with building clinical evidence I will cover some of the methodologies. Basically, what we're calling it is virtual patients, and so we'll talk a little bit of how we build virtual patients in general, and then talk about how we leverage those as priors in a Bayesian clinical trial. And then we'll hand it over to Adam to actually talk about the real world examples that we had actually incorporating these methods into real clinical studies at Medtronic or in the medical device industry at large. Next slide, please. So the medical device industry has a real problem. If you look at the past, the present, and future, we're having real issues with the cost of our clinical evidence, building our clinical evidence. We're having increasing demands of having longer duration clinical trials and larger follow-ups, just actually having more more sample size, so requiring a lot more patients, and then also collecting more information about each patient. So really, collecting so many different imaging and demographics and medication, and it's just altogether it's increasing the cost, and this trajectory just keeps going up. And so this was identified a while back as something that has to change in order for the medical device space to really continue to innovate, because clinical studies are really the lifeblood of medical devices. And so if this cost trajectory continues, that's going to stifle innovation in the medical device space and really prevent life-saving therapies from getting to the patients who need them the most. And so, next slide. So this is where, we thought about it and we said, there's real opportunity for bending this curve. And what we realized when we looked at the problem is there was a tremendous amount of information that you could derive about the therapy that you were looking at from other sources other than just the present clinical trial that you were running. So we had previous clinical studies. So a lot of the products we have some sort of predicate device. It might not be exactly the same, but it has some sort of relationship that gives you a lot of information about what that device's function is, what its performance would be. We also have a lot of real world data. So sometimes there is off-label uses of some products that are now trying to become on-label. That information can be really relevant for information about your present clinical study. And then probably one of the biggest areas of advancements in the last 20 years has really been about engineering model or numerical modeling. Everyone has heard the term like digital twin and all of these advanced methodologies to really get at how a device can interact with the body, and those methodologies are getting incredibly sophisticated, and that information can definitely help you to be informed with your current clinical study. And also there's Bayesian statistical methods, and this is really an incredible way of incorporating the above information I just discussed in a way that helps you to bolster your existing clinical trial and reduce some of those burdens around size and duration. And that's really what we're going to be talking about today, is how can we leverage the above information, and particularly we're going to focus on engineering models and how they can be leveraged as a prior in a Bayesian clinical trial to really bend this curve. Next slide, please. So we're going to talk a little bit about the methodology. You can just go to the next slide. So when you think about, traditional sources of information, it really is broken up into these main categories. When you're building a medical device, you kind of start, with maybe some bench testing. Maybe you'll do some sort of understanding. You'll replicate the anatomy and the use conditions in some sort of a bench test just to get a flavor for things. or you'll understand the reliability of the device. You'll generally do some animal studies to understand that, and you might do some computational modeling to understand the fatigue performance or the electrical performance of it. But you can see then, a lot of it is human data, right? A big portion of the pie is humans, and that's obviously the most expensive part, and the part that subjects humans to untested products. So, if at all possible, it's good to minimize the amount of exposure humans have, both from a cost perspective and from a human perspective, and capitalize as much as possible on the other three areas. But there's really kind of a weakness inherently in both animals, bench, and computers. So next slide. And that's really where we came up with this idea of the virtual patient. And the virtual patient is really, combining all of these methods together to build a virtual patient, and that allows you to reduce the total reliance on humans and cut that human pie a little bit smaller, increasing the computational and the virtual patient side. So that's been a huge trend in the medical device industry, to really, really spend a lot more effort on understanding your products and understanding the humans in more of a computational virtual world, through understanding physiology and those kind of things. And so when you combine these physiological and numerical modelings with some probabilistics, and we'll discuss that next slide, then we can incorporate those into the clinical trials that we're trying to build through Bayesian methods, which really is a very good way of incorporating this information, and very analogous to why Bayesian methods exist. Next slide. So what is a virtual patient in general? So think of it as it's three fundamental components, right? The first component is, physiology, physics models or physiology models. And that's really, if you're talking about a medical device, you could do the same analogy with like a pharmaceutical company, right? But in this particular case, we're focused on the medical device space. So really it starts with, mechanically understanding or electrically understanding, or understanding your device itself. So that's really the mechanics, the electricals, the heating, whatever the device is doing, whatever its form factor is, understanding that device in a very, very, very solid sense, mathematically, numerically, and simulation-wise. The next really important area is the physiology. So this is really understanding the biology of the human and being able to model that. So as an example, we have the cardiac multi-physics model. So this is like understanding things like hemodynamics and electrical systems of the heart. And 30 or 40 years ago, we'd be like, well, how can you do that? But now there's incredible models that actually can simulate hearts, right? Virtual hearts, and understand that if you put, let's say a pacemaker in a certain place, you can actually understand how those hemodynamics of that heart change, and how it even can remodel due to that. So it's really about understanding that biological response to that device. Then we move on to probabilistic modeling. So it's not enough, what we saw, Adam and I and some others a few years ago, is they were doing incredible things with these numerical modelings, but they weren't really doing it in a statistical probabilistic way. So they would, replicate one heart, some particular heart. They would do some imaging and they would replicate it, and they'd say, look, if you put a device in this one heart, then we can simulate it. But that's not the way a clinical trial works, right? You've got natural variation. You've got different ages, genders, anatomy types, organ size, different diseases, different indications. So all of those things really contribute to the heterogeneity of patients that are implanted with a particular device. let alone the device itself has some level of heterogeneity, right? In things like fatigue and the strength of a device, if you're worried about the reliability of a product, there's going to be natural variations in the material. And so all of that stuff is encapsulated in a clinical trial. So if you want to create a virtual patient, you have to encapsulate those same elements, right? So you have to vary the anatomy, vary the size of the organs. You have to vary the disease state. You have to vary the activity levels of that patient, implant factors, lots of variability going into that area. And the other thing that you have to deal with is the uncertainty, right? So let's say you're building, let's say you are understanding your device and you do some bench testing, you do it on, let's say, 15 samples. Well, and you're trying to understand fatigue. Well, there's a sample size associated with that, right? So your fatigue performance is going to not be a fixed parameter. If you say, oh, the fatigue is normally distributed with a certain mean and a certain standard deviation, well, that mean and standard deviation was estimated by a sample size. So you have to account for that uncertainty. And same with measurement error. All of these things are using measurements, and so some of those have measurement errors. So that's that building the uncertainty in your model as well. And then the final step is really taking all of this information and bringing it to a clinically relevant outcome. And I think this is a really big step and actually probably one of the most challenging steps, but it's not enough just to be able to model that, all of this activity. You have to be able to construct some clinical endpoint that's replicating the clinical endpoint of your clinical trial. So in this case, which Adam will discuss, we were going to talk about our cardiac pacemaker leads or defibrillation leads and their failure rates. So you have to bring it all the way to a point where you can literally simulate a survival curve of those virtual patients. Same with like blood pressure or glucose values. You have to be able to take it all the way to what a clinical endpoint would look at, which might be the percentage of patients that are reducing their blood pressure or maintaining their glucose level. So it can be challenging, and that's really that final step is what is necessary to be able to use this in a virtual patient clinical trial in a prior. Next slide, thank you. So really at a high level, what are we trying to do? We're trying to, reduce the amount of clinical evidence as we have more maturing models. And so really this replacement can come in phases, right? So at the beginning, and this is why a Bayesian clinical trial really lends itself well, because you can basically increase the strength of the prior as your information, as your ability to develop these virtual patients strengthens. So at the beginning, you might only use a little bit of virtual patients in your clinical evidence, right? In some cases, you might be to the point where you're not even going to put it in your trial. It's just enough to get you comfortable with running a clinical trial on humans, right? That's sort of that first version where you go to FDA or any regulatory body and you say, hey, we've done this, numerical modeling, we've done these things, we understand it's enough to be comfortable moving to a clinical study. But as that numerical model and those virtual patients become stronger and stronger with incorporating the things we just discussed, you can start to say, we don't need to run as big a clinical trials. We can make these trials smaller and smaller and smaller and just have them finally be very much confirmatory of what we basically already know from our virtual patients. And so that allows you to really reduce that clinical evidence burden and as the model maturity grows. Next slide. But as a biostatistician, one of the really important things, and the things that I always had trouble with Bayesian trials, was the fact that you have a prior, right? And that's in principle, the prior is supposed to be something that is unchangeable, right? But in reality, what we found is when we looked at a lot of the methodologies, there was a tendency for your type 1 error to be really, really big and your bias to be extremely bad in your estimations, even when there was a lot of statistical evidence that your virtual patient model was wrong. And that was the point where, even though, in statistical principles, it's like, okay, that's your prior, and it's going to slowly convert to the likelihood. Your posterior will force it closer to the likelihood. At a certain point, we realized that it isn't moving fast enough, right? That we needed to nudge the likelihood to be more based off of the likelihood and less based off of the prior, when there was a lot of statistical evidence that you just made a mistake. And this is really kind of the point of a clinical trial a clinical trial is designed to make the right decision with the most efficient amount of use of your data. And so what we started to think about, and we worked with FDA and MDIC, which Adam will discuss what MDIC is. How can we make a methodology that allows us to leverage the prior to a certain degree, but then at a certain point, that if there's too much disagreement between the prior and the study data or your likelihood, that you would essentially start to discount the prior in a more rigorous way. And this really has the effect of trying to maintain that statistical power when things do agree, but really reduce that type 1 error and that bias that you can see if things really go off the rails. And so that is the discount prior approach, which I'll discuss now. Next slide. So here is what we're calling the discount prior approach, and I'll kind of go through it step by step. And you can see the methodology in a paper we published in 2017 in biopharmaceutical statistics. So the first step is really to set, and this is all a very pre-specified approach, and I think that's really critical for maintaining type 1 error. You have to pre-specify this whole process. So the first step is really to set your maximum weight of your virtual patients or your prior. And by the way, this methodology can work in any sort of a Bayesian setting, even if it's just a historical data set, historical data that you want to use as a prior. So you set some max n. So in this case, let's say 50% of the weight is coming from our historical data or our virtual patients, and that's the max. So it can only go down from there. And so then when you have your clinical data and you have your historical data, in this case we're saying our simulated patient data, we then look at the overlap, so a statistical comparison between the two, and that agreement A is really the amount of agreement that exists. And so you can see that this is like the statistical agreement between the two. And if there's less overlap, then you get less weight for your clinical trial. And it can be as simple as this linear relationship, right? So if there's 100% agreement, then you get 100% of the weight, or in this case, 50% of the virtual patients. And then as that probability of those two being from the same distribution decreases all the way down to 0, you get 0 weight. And that's what this is called, the discount function. And then you do a simple calculation with you define what your prior is with this power prior approach, which is applying the power prior, which we won't cover, but it's a well-known methodology for discounting your prior data. And then you can then calculate your posterior distribution from this approach. Next slide. So a few years ago, we actually did some simulations to try to understand how this discount prior approach compared to some, traditional methods like hierarchical models and power prior, traditional fixed power priors. Or not a fixed power prior, but it was like a hierarchical power prior approach. And then a commensurate prior approach is another approach that is in the literature. And what we found is, so this is a bias. So this is just a simulation. If we assume that the historical data or prior is set at 0 with some standard normal distribution. And then the true data varies from that prior. We can see the profile of some of these methods, right? So if there isn't a huge difference between the true value and the prior data, in all of the cases, you can see this is exactly what you want to happen with the Bayesian method, right? So you can see you get strength from your prior. So you see a little bias, but your prior is holding on and saying, I don't believe that. I think it's a combination, and so it's kind of like a weighted average. But what's important to note is some of these methods, you can see that even as, so this is in terms of like, sigmas, right? So standard deviation. So as you see sometimes as the true data or the truth gets really far away from the prior, in cases like where it's statistically significant, and we call this the discordant regions. So this is where it's clearly different, right? It's a 1.5 sigma away from where your prior is. There's starting to become a huge amount of evidence that what you thought was the truth is really not the truth anymore. And so you can see that the other methods, by the way, I think you can just worry about the solid lines. The dotted lines are just some other some other discount priors that we were choosing. The one on the top is just that linear relationship, just that the probability of overlap is used as the actual, the line. And that one has very good operating characteristics. You can see that some of these other historical methodologies continue to hold on, right? They continue to hold on and adding bias to the model, even though there's clear, undisputed evidence that the prior was just an erroneous prior. Whereas the discount prior, you can see that it holds on, but then at a certain point when there is true statistical evidence, it lets go. It says, you know what, I made a mistake, and now I'm going to just fully focus on the likelihood or the existing current data to make a decision. And so I think what we found was this is a very good feature, right? So you leverage a prior when there's some ambiguity, and that you could theoretically have that prior be useful, but then at a certain point you just have to let go, because any regulatory body, any statistician is going to be like, you guys made a mistake, and so we don't trust the results of your endpoint. And so that's kind of what we show here is when there's a p-value of 0.001 that the prior and the current data don't match, this creates a bias in the direction for keeping the treatment, and that just adds distrust to the clinical study as a whole. So that's why the discount prior seems to work well. And I'll not cover this too much, but we saw a similar situation with the type 1 error. When there's a statistical difference, you can see the type 1 error as you're far away from the prior, that the other methods maintain that bad type 1 error. So it continues to leverage and ruins your type 1 error across this discordant region. But for the discount prior, the type 1 error starts to converge as there's a lot of evidence that the prior was wrong. It starts to converge to a nominal type 1 error of 0.025. So that's the discount prior. I think next slide. And with that, I'll hand it over to Adam.
All right, thank you, Tarek. So just as by way of a quick review, we introduced the problem statement that the clinical evidence, as important as it is getting increasingly challenging to generate with rising costs really going up exponentially. We've shown a method where you can take prior information, whether it's from engineering models or other sources, and incorporate it using Bayesian statistics. Bayesian statistics aren't necessarily new, but the idea of this discounting method to really put a lot of scrutiny on how well the prior agrees with the current data is something which is relatively novel and hadn't been done a lot before. The next step is putting this into practice. And this is where there's a lot of collaboration needed across the many stakeholders. And for the purposes of this example, we're going to talk about collaboration with the US FDA. Certainly, there's many geographies and other regulatory bodies. For the development that we are doing here, we are able to have some direct collaboration with the US regulators. So I'll start there, and then I'll get into examples of what this collaboration looked like in specific terms. And then we'll talk about actual implementation in a recent clinical study that was run by Medtronic. I'm going to introduce our device example here. It's always easier to think about these things when you can have an actual product in mind. And what we'll be talking about is implantable defibrillator leads, or ICD leads. This is a really quick introduction. An implantable cardiovascular defibrillator, or an ICD, is a device that's permanently implanted that's able to detect life-threatening rhythms and then deliver a shock that can restore normal heartbeats. So this device operates a lot like what you might see on a medical drama, where the patient is having a cardiac arrest and the doctor says clear and the paddles are applied and then a shock is delivered and then the patient's heart starts beating. You see the electrogram resume normal activity. That's really exactly what an implantable ICD does, except that the difference is these implantable devices are something that people carry with them everywhere. The function of the ICD lead is to deliver the energy from the pulse generator that's implanted, usually in the shoulder region, through conductive wires to a location within the heart. So you can imagine now the importance of these wires. If they break, there's many failure modes that can happen, and they're all serious. It can range from inappropriately detecting rhythms. So if the wire is broken and it's kind of making like make-break connections, noise, then the device might think that you're having ventricular fibrillation and you're not, and deliver a shock. And then if it's broken altogether, you might need a shock, but the device does not detect it or cannot deliver it, and then you fail to receive that shock. And obviously, that's got life-threatening consequences. So fracture of these conductor wires is a very important failure mode. And for this particular example, the thing that makes it most relevant is that this particular failure mode had been driving the size and duration of clinical studies. So there's a device, a particular failure mode, and a clinical implication. And that really kind of set the stage for what we did in terms of our collaboration and then the actual study. So obviously, there continues to be a need for innovation. Manufacturers are always looking for ways to make things more reliable, have more features, et cetera. But in order to do this, we need to generate the clinical evidence. We want to be able to do this as efficiently as possible and do it in a predictable way. This is important not just for the manufacturers, but also for the regulators too. There's many different products that the regulators have got to evaluate. So this makes a good example here. It's relevant in terms of something that regulators and manufacturers are actively having to deal with. The particular failure mechanism of conductor fracture is not complex from the standpoint of all the biological things that can happen. It uses a lot of the same mechanics that are well studied in many other industries, from transportation. Think about airplane wings and making sure cracks don't happen there, maritime ships, power generation, even things from elevator shafts. All of these have flexible metal structures that have to be able to resist fatigue. So there's a good body of evidence from other industries. Also, there's published examples. So we had publications specific to this model that were already in the public domain. So it's a lot easier to collaborate with people outside your firm when you can work in a public space without having to get into confidential information. I'll talk in real broad strokes of what this engineering model looks like for ICD lead fracture. To get more details, you can look at the paper that we published. But you can kind of think about this as two different sets of input data. There's use condition data, or what is the device actually experience, and then there's strength data in terms of how much use can the device withstand before it breaks. The use condition data, when we're talking about ICD leads, is really two different aspects. One of them is how much the lead bends, and it's really as simple as that. You can think of any kind of flexible structure. Think about your phone charger cables or perhaps earbud cables, and we've all had those break in the past. A lot of it started with how much that was breaking. You might realize, like, oh yeah, I put these cables into my pocket, and it was kinking in a certain way every single time, and that's why it broke. Well, the same sort of measurement can be done on these implantable ICD leads. So there's how much it bends, and then there's how many times it bends. So you can think about your own examples, whether it's charger cables, or we've all maybe bent paper clips either in a science experiment or out of if you're just like sitting at a desk and you flexed a paper clip too many times back and forth, and after so many cycles, 10, 20, 100 cycles, then it fractured. It's that same number of cycles that is relevant when we're talking about fracture of these conductors in the ICD leads. So those are the use conditions, how much it's bending and how many times it's bending. The strength part of this equation is taking leads and subjecting them to bending cycles at increasingly tight radii or high stress levels, and then measuring to see how strong they are. So you think about the implanted use condition that's going to bend a certain amount of times. We can do that same sort of bending on a bench and take a measurement to see, well, if it's bending this much, how many millions or hundreds of millions of cycles might it last? So with those data sets together, the use conditions and the strength, that's the ingredients that we need to do Monte Carlo simulations of an actual simulated patient population. So we can draw examples from distributions for all three of these inputs. In patient A lead might bend by a certain amount, and they might have a certain activity level. So we know that every year, here's how many million cycles the lead is going to experience. Then we'll have our collection of bench test data where we've tested at many different kind of stress levels. And we know that at the stress level for patient A, here's how many million cycles it might last. So then we can simulate the outcome for that patient to say that a fracture would occur in 8 years. And we repeat this over and over and over again. And hopefully then you can appreciate that with these distributions for all the sources, we can essentially simulate a clinical trial in a virtual population. If you remember the equation that Tarek showed a few slides ago, where you had your physical models, probabilistic models, and a clinically relevant output, the clinically relevant output here is fracture of the conductors in the lead. One of the key things about these devices is that they're actually able to measure the electrical properties of the lead conductors. So what that allows it to do is the device can measure exactly the same thing that we measure in our bench tests. And the failure mode that happens clinically, fracture of the conductor, can actually be detected by the device, just like we can detect it on the bench. So we're measuring exactly the same thing in our bench tests as would be measured in a clinical study. So our outcomes are entirely exchangeable. And this is really table stakes when we're talking about having an engineering model that can be used as a prior when we're analyzing clinical study data. We have to be simulating an outcome that you would actually measure in the clinical study, and that's what we're doing here. So we've developed this modeling framework, and this is something that was primarily developed internally, drawing on experience from other industries, but the broad strokes of the model were something that we developed as we were looking at performance of our own leads. These methodologies, though, were different than what had been done before, and when we think about incorporating it into a clinical study, that's extremely different than anything that had been done before. So anytime you're doing something different, regardless of who your stakeholders are, there's always going to be questions, and it's always going to be disruptive. When it comes to introducing new devices, new medical devices, predictability is really important for all stakeholders involved. And the risk of delays due to long feedback loops, so you can think of you're doing something brand new and you're going to go and talk to a regulator about it, there's certainly going to be questions. We really wanted to minimize the question and answer period that would occur during the regulatory submission process, so that we would know that when we're all done with our work, then it'd be a very straightforward review that regulators could do. If there is too much of a barrier here, then a firm might decide to not do this innovative new approach. So you can kind of see that there's this negative feedback loop that could occur if you're too different. The priorities of industry and regulators, as well as physicians, are in many ways are actually really similar. All parties are really interested in bringing new products and therapies that are more safe and more effective than the previous ones to market, so that outcomes can be better for patients. That's something that we all share. So we're looking for ways to do that. And this is where having these frameworks like the Medical Device Innovation Consortium come into play. So MDIC, our Medical Device Innovation Consortium, is a public-private partnership that gives a space for regulators, so government agencies, for industry, and for nonprofits. This could include providers, patients, advocacy groups, to all work together on various projects. You can go to the website mdic.org and see numerous different projects where MDIC is giving a space in the pre-competitive area, meaning this is not jointly developing products together, but really developing methods together. One of the projects is computer modeling and simulation, and we kind of lived under that one. So this was a really key step. The first collaboration was publication. So when you're introducing a new method, one of the best ways to get it socialized, it really is through the publication process, especially if you have joint authors from different agencies. And that's what our publication represents here. There's authors from both Medtronic as well as the US FDA working together to develop the exact statistical methods for incorporating engineering data into a clinical study using this discount function approach. This is open access. It's available online in the Journal of Biopharmaceutical Statistics, and it's definitely worth a read to see the fine details of what we're doing. The timeline here is not unusual to folks that have been through a publication process, especially if it's on some kind of new idea. One of the reasons I put this in here, though, is that to highlight a theme that comes up, just like you would expect with any disruptive thing, that when you're doing something that's different to the status quo, and in this case, it's introducing engineering data into clinical study analysis, that's something that folks have a variety of comfort levels with, I think I would say. And for journal number one, the comfort level was uncomfortable. So it took going to a second journal that was a little bit more open to innovative methods for clinical study analysis approaches. And even then, there was definitely a lot of back and forth with the reviewers, but ultimately we got it published online. The second collaboration, and now we're getting a lot closer to putting these ideas into practice, was a mock submission. So this is just like it sounds. We went through the steps of submitting a device or a clinical study application to FDA for a hypothetical device in a hypothetical study. And this was really important so that we could identify what were going to be the sticking points or the hurdles or the sorts of questions that might arise. So the way that this worked was we had a sponsor team. So this was a hypothetical firm. And it was actually composed of people from different companies around the medical device industry, as well as representatives from FDA that we worked together. We described this clinical study and our hypothetical new device that we were going to investigate. And then there was a review team. So FDA actually set aside time for employees to participate in the review of this mock submission. The documents here are available on the MDIC website. It's definitely an interesting read, worth looking at. So we developed a hypothetical new ICD lead. I won't dwell on the actual construction, but suffice to say that this was a kind of like a synthesis of what the leads look like across the industry. And we proposed a possible change that is plausible, something that a company might do that would be significant enough that it would likely require a clinical study. We developed a relatively straightforward Bayesian adaptive study to look at what the safety outcomes would be. So since this was specifically targeted at the use of engineering models in the submission, we didn't want to design a study here that would look at every single possible thing. We were really focused just on that safety objective for conductor fracture. We had a hypothetical fracture rate at 18 months that we'd expect to be less than 1%. We had performance criteria that it had to be better than 3%. And this is in line with what we'd seen for predicate studies. We had an enrollment process that would have stopping criteria for success. So we would certainly want to stop early if it was indisputably clear that this device was exceeding its performance criteria, as well as for futility. If it was very clear that this device actually had safety concerns, we want the ability to stop early and not subject additional patients to risk. And we looked at 4 different scenarios. One would be a traditional study with no actual prior information whatsoever. We looked at the fixed prior, meaning you're going to do a Bayesian study, bring in the prior information, but that weight would be fixed no matter what the level of agreement would be. And then a couple of different discount function scenarios. One was more conservative and one less conservative. So we can look at the results here, and there's a lot going on, but we'll talk through it. So to start out with, if you look at a traditional study with no virtual patients, that's shown in the gray line kind of on the left in the graph, this was underpowered, meaning that even though we would likely be able to detect a dangerous device, we had so many false positives because the sample size was relatively small. So no firm would likely undertake a study that only had 60% power. There's too high of a chance of failing, even if your device was perfectly safe, just due to a false positive result. If we look at the case with a fixed prior, and this is what a lot of traditional Bayesian studies actually suffer from. So if you look at the evidence from medical device Bayesian studies, this is why there is really a lot of skepticism, because in the setting of a fixed prior, if you're wrong, if you're optimistic, and oftentimes firms can be optimistic, and your device or your prior information is actually better performing than your actual clinical information, the type 1 error is unacceptable, meaning that you might actually have a device that has got safety issues, you don't meet your safety objective, but because your prior is too optimistic, it's actually pulling it up, and you might pass a device that should not pass. And this is where the discount prior really comes into play and shows the benefit of an approach where you're adjusting the weight of your prior information according to the level of agreement. So in the cases where your prior information, in this case, our engineering model, agreed well with the clinical data, we have just about the same study power, or just about the same % chance of passing, which you should. But the really important thing is if you look at the cases where the engineering model is wrong, then the type 1 error is just about the same as it would be with no virtual patients at all. So it shows that the approach is working as desired. We're bringing in our prior information to give us the chance to pass the study when our prior is correct. But if our prior is wrong, it gets deweighted, and you could basically convert to a traditional study. So this is doing all the things that we would want it to. So the timeline here was I went through about a 3-year process. There was a lot of work that was done beforehand, and then next I'll speak about the work that was done afterwards. Well, we learned a lot during this time frame. Model credibility is the #1 issue here. So we need to make sure that our model is actually believable, that it's worth actually bringing this model to replace human data. There's a lot of stakeholders that are going to be looking at different things that they're not used to. From an engineering perspective, it's unusual to have biostatisticians looking at an engineering model. From a biostatistician's perspective, it's unusual to have engineering reviewers that are actually looking at it. So all parties need to be prepared to speak different scientific languages. So let's get into actual application. So we went through building up the biostatistical method. We went through the hypothetical example of what you might do with such an engineering model. Now let's talk about a real example. So this is an actual study that was run. On the bottom left, you can actually see the clinicaltrials.gov information. Medtronic recently released a new ICD lead. The details aren't really important for this particular presentation, but the high-level attributes here are this was a lead that was designed to have extremely high reliability. It was based on an existing platform that already had a lot of data around it. So we weren't doing something entirely new, but rather we were able to look at what was done before and show that the adjustments that we were making were in the positive direction. And then we had a clinical study design where the thing that was driving the size and duration of the study was conductor fracture of the wires inside the lead. So kind of following the footsteps of the example that we presented here, we actually have a product that meets all the criteria for what we demonstrated before. So in this case, we have an actual study where at least 500 patients were going to be enrolled, followed for at least 18 months. And we had agreement that we were going to have up to 500 simulated patients. The secondary objective was to estimate fracture-free survival. The step-by-step process here that Tarek showed earlier is one that we wrote directly into our clinical study plan. So we had our maximum weight. So we weren't going to allow the virtual patients to be any more than 50% maximum of the total data set. We had our pre-specified steps where we're going to compare the clinical and actual outcomes. We're going to look to see what level of agreement there were, and then we would actually do calculations on that combined data set. Here's a look at the actual measurements that were taken. So when we talk about these use conditions, hopefully the videos are coming through in the animation here. If they're not, you can certainly reach out to me and I can send you these slides as well. These red lines that you see in the videos here are actual tracings of this ICD lead bending and flexing inside patients as they move their arms around and the hearts beat under a cinefluoroscopy X-ray system. So this is measuring the exact things that are causing the stress to be applied to the leads. And we apply similar stresses to the leads in our bench tests. So we're actually simulating what could happen in the clinical environment, doing this so that we have distributions that we can conduct Monte Carlo simulations from. These are the clinical study results. So this is actually a slide that was presented at the Heart Rhythm Scientific Sessions by the clinical study principal investigator, showing that we had our study cohort. In this case, there ended up being 650 patients implanted. The reason why there's more than 500 is that it's hard to stop a clinical study on a dime, and so the enrollment period does continue a bit. We were able to include 242 simulated virtual patients. The reason why it's not exactly 500 is because the clinical performance is actually outperforming our virtual patient data. And we had a very conservative discount function where if there's disagreement, either positive or negative, we were going to de-weight. This allowed us, though, to compute a secondary safety objective of a 99.97% 12-month fracture-free rate. So you can hopefully appreciate that this level of precision is not possible with only hundreds of patients. You need to have the benefit of the engineering model to get you down to the lower decimal points. So the key concept here is that the clinical data and the model together allowed us to have a much more precise estimate for reliability. The real value, though, comes after once we've done this first level of demonstration. This essentially has validated our model against the clinical data. And then there's additional applications. And we can just kind of roll down through these. You can look at what would the performance be in a different patient population. In this case, adolescent pediatrics, where for teenagers that would receive this lead, that would have a higher activity level, what those outcomes might be. We can look at longer-term follow-up. So the value of a model is that you can go beyond what the clinical timeline actually is and make predictions for, in this case, it was up to 10 years. You can look at new implant locations, where if the doctors are going to implant this lead in a different shape that might change the use conditions, what would the performance be in those cases? So we've been able to look at each one of these, and in each case, it's actually been published as well. So I'll wrap up and summarize. We've gone through the approach of using virtual patient data in a clinical study evaluation, and this provides a lot more efficiency when we have the right model for the right clinical data. Some of the key steps along the way were early collaboration with our stakeholders, especially regulatory stakeholders, due to the difference of this approach. Having the model that's developed for the right clinical objective is crucially important here. If you had a model that was developed for something that was not driving sample size and duration, it would be interesting, but it wouldn't be able to make a large effect on the actual clinical study. And the real success here is beyond the engineering methods. This allows us to get products with good performance out to the market faster than we'd be able to otherwise because of the efficiency that was gained with this approach. We had a clinical study that was more economical. We were able to enroll faster and end faster. And the result is a product which is currently being implanted across the world. All right. With that, if there's any questions, Tarek and I'd be glad to answer them in the next 10 minutes.
I think the value of artificial intelligence could be in improvements in the development of the virtual patients. I think that leveraging, particularly, there's a lot of efforts in numerical modeling to help with some of those physiological things using machine learning and AI techniques to improve those virtual patients. I think there's a huge opportunity there. And I think that, I think the whole digital twin concept, I think, can lend itself. Imagine you have 10,000, let's say in the future, where everyone has this really incredible digital twin, they could sign their digital twin up to be part of a clinical study, a virtual clinical study. So I do think that that could be something where we're all deciding to enroll our own digital twins in all these different trials. Kind of maybe far-fetched, but could happen.
I can add on to that a little bit. I think kind of back to the lessons that we learned along the way. As those opportunities or areas where you would have a digital twin enabled are arising, it's really important to start as early as we can to understand what's the fidelity of the model that needs to be developed. get that in place ahead of time. If we learn one thing from this approach, I think Tarek mentioned the 10-year time period, and it really was about 10 years from when we started working on just developing the framework for the model to when we are actually analyzing the clinical study results. So that's quite a long time. We don't want to wait 10 years for the next innovation, but these things do take time to work together on.
Yeah, I don't think there's really much difference. I think as if the pharmaceutical companies have their virtual patients, their physiological models that understand the chemical reactions and how the body interfaces with the pharmaceutical, I think it's completely akin. I don't think there's much of a difference. So I suspect, not being in the pharmaceutical industry, I suspect they're doing this type of stuff quite a lot.
If we think about examples, again, just as Tarek's not in the pharmaceutical industry, neither am I, but we think about dosing. So certainly comparing like one drug to another drug, many different things could be going on. But if we were to look at perhaps changing a dose of a drug where you have like the same biochemical reactions happening on, but maybe to a different magnitude, or perhaps looking at expanding indications to a different patient population. That's where this approach could be brought to bear, where especially the value of the discount prior is when there's agreement, the study and the discount prior method proves that agreement. And when there's disagreement, that also comes out there as well too. So you get the valuable statistical benefits that we showed earlier, but then you also get the validation or rejection of your model. Yeah, the adolescent pediatric example is a real one, and that's a particular challenge that is well recognized, that enrolling pediatric patients into clinical studies is something that it's done very seriously, just because it's a vulnerable population. They cannot consent for themselves. And when you're talking about children, obviously it's a very compassionate patient population. Oftentimes, diseases affecting the pediatric population are rare, which makes enrolling a clinical study even more difficult. So for this particular application, we looked at data from a number of different sources, and it turned out to be that the key thing for getting labeling for adolescent pediatrics was really the body habitus and movement patterns. So we did a couple of things there. Is we were able to look within our clinical study to see how many of our adult patients actually could be close to adolescent pediatric body habitus. So just think of small, more active adults. And in our bench testing, we looked at those outcomes in the bench tests where we were doing much more stressful tests of the ICD leads. So while we weren't able to actually, in fact, we did not even try to have a distinct adolescent clinical study. We were able to look at the characteristics of adolescents that would be relevant for the clinical outcomes that we studied. So it really ends up being, I think, case by case when you're looking at these hard to reach populations. But it's a great example of it.
Yeah, I think if you can create those transfer functions, then the world is your oyster for using these methods. It's all about building the evidence that there's similarities, and if you can convince yourself and everyone else that the animal data is similar enough to the humans that you can build that connection, then I think the methods could be used for all of those use cases. But you have to verify that in a make those connections. And I know we've struggled with that at Medtronic, of making connections with seeing things happen in swine models and not seeing it happen in human models, and vice versa. So and all sorts of things. So there isn't a perfect translation, but usually it's directionally correct. Thank you.
Thank you, everyone.